Armand was introducing me, you've probably all been staring at this picture on the screen of these ants. And you might have noticed that some of these ants don't look exactly the same as the rest of the ants in this picture. So if you look at this little one down here, its abdomen is slightly yellowish, right? Its anterior body is a slightly different shape from this one over here, right? That's because this ant isn't actually an ant at all. It's a beetle that lives symbiotically with these ants, and is accepted by them as if it were a nestmate, which sounds really kind of harmonious, except for the fact that this beetle has infiltrated the ant colony and lives amongst these ants as a freeloading parasite that feeds on the ant's own offspring as a socially parasitic organism. And what I'm going to talk to you today about is my lab's work on these beetles and this way of life, and how it sheds really kind of fundamental light on a feature of the biological world that's so pervasive, but we know very little about, which is the ecological relationships that exist between different species. Now, I would say ever since Darwin, we've developed a pretty good understanding about how evolution works at the level of individual species. So, you know, from the kind of level of the nucleotide and the gene all the way to the population, how individual species emerge and evolve and speciate and diversify. That's something we know a decent amount about. But what we really don't understand in real mechanistic detail is how interrelationships between different species evolve, right? Species are really nothing without their interactions with other species in the biosphere. But what it is that enables species to form nascent relationships with each other, how they fall into new kinds of interactions with each other, how those interactions undergo evolutionary specialization, leading to things like symbioses, and much more specialized kinds of interactions. This is not something that modern biology knows a huge amount about. And so one of the problems has been our kind of focus on a small number of model species like fruit flies and mice, which don't really show interesting interactions with other species. They're very easy to keep in the laboratory. They've illuminated really kind of fundamental mechanisms at the molecular and cellular level for many different biological processes. But when it comes to understanding how these organisms interact in an ecological context, they've not yielded very much. And on the flip side, things like, you know, the symbiosis between a remora and a whale shark, well, that's basically impossible to reconstitute in a laboratory and study in mechanistic detail. And consequently, our understanding of the biosphere and the connectivity between species is still in its infancy. And so what my lab is interested in doing is trying to understand this kind of evolved architecture that connects different species in the biosphere. And the group of organisms that we use to study this phenomenon are the beetles. Now, 1 in every 4 living things that humans have given a name to is a species of beetle. There's about 400,000 described species of beetle. It's, you know, the largest group of organisms on the planet. But you have to wonder why this kind of thing is. Why are there so many beetles, right? And it's long inspired evolutionary biologists to ponder the diversity of life. You know, Charles Darwin and Alfred Russel Wallace both started out their kind of biological careers as amateur beetle collectors. And the diversity of beetles is also behind evolutionary biology's arguably most famous quote, which comes from J.B.S. Haldane, who was asked by a theologian what you can infer about the mind of God from studying the natural world. And Haldane supposedly replied that God surely has an inordinate fondness for beetles because there's so many of them. So why are there so many beetles? Well, I think the key innovation that beetles have that other organisms and other insects don't have probably is the recipe for their success. And this innovation is a structure called the elytron. So if you look at this video of this beetle here, you can see this hardened shield-like structure that protects the abdomen and the delicate flight wings. This is called the wing case or the elytron. And it protects the beetle's ability to fly. So the beetle has flight wings, it can still fly around, but this structure covers those delicate flight wings and physically protects the entire body. And what this means is that beetles are able to infiltrate many different parts of terrestrial ecosystems that are shut off to other insect groups that have delicate membranous wings without these wing cases. Okay, so beetles can burrow down into the ground, they can live under bark, they can infiltrate many different internal parts of plants, they can live inside dead wood. All of these habitats are really shut off to other groups of insects, and consequently, beetles have speciated and diversified in many different niches that other insects can't get into, but they can still fly and disperse and speciate. And this is probably why there's about 400,000 known species of beetle. Now, what a lot of people don't realize is that the largest group of beetles, so within the order Coleoptera, the largest group of beetles is a kind of beetle that doesn't look like a typical beetle. It's a group called the Rove Beetles, this family Staphylinidae. Okay, and that's going to be the subject of today's talk. Rove beetles are a huge radiation of beetles, 64,000 species, about the same species richness as all of vertebrates put together. And they don't look like your typical beetles because they're elytra, their wing cases are very short. Okay, you can see these structures here, and they actually expose the abdomen. So you can see all of these segments here. These are the abdominal segments. And the abdomen is kind of flexible and the segments telescope together. And that enables rove beetles to move rapidly through substrates like soil and leaf litter, okay, chasing down other invertebrates, which they feed on. So within the beetles, what you have to kind of imagine is that there's been a secondary modification of this physically protected body plan, where the elytra have become shortened, enabling body flexibility in these beetles to move rapidly through substrates, right? And these beetles have exploded globally in diversity as this kind of predatory group. Now, this is a particularly fancy rove beetle. Most of them do not look like that. Most of them look like this, okay? They're kind of dull, brown, black coloration, you know, easily kind of overlooked by biologists historically because they're so small and incredibly species rich, okay? But they are exceptional not only because of their incredible species richness, but also for the reason that many times during their evolution, they've gained the capacity to infiltrate a type of habitat which other groups of insects, including other beetles, have not been able to get into, which is the ant colony and colonies of other social insects. Now, most insects that would wander into an ant colony like this poor beetle here, this red guy here, you can see it's recognized by this ant instantly and attacked by it, okay? So ants have on their body surface these chemicals called cuticular hydrocarbons that they secrete onto the body surface, and they encode a signature of colony identity. So ants recognize members of their own colony based on the similarity of their cuticular hydrocarbon or CHC profiles. And what's happening in this video here is that this beetle has CHCs on its body that don't match those of the ant. And ants are innately programmed to be aggressive towards things that do not match their own chemical template. Okay, and this is the reason that ant colonies are really ruthlessly policed against intruders and have this social cohesion. Okay, it's this chemical identity that ants use to recognize members of their own colony. What rove beetles have done hundreds of times during their evolutionary history is evolve strategies to bypass or hijack ant nestmate recognition systems. So if you look at this brown beetle down here, far from being attacked by that ant, it's actually being fed mouth to mouth by it. Okay, so that ant is regurgitating liquid food to this beetle and is feeding the beetle, right? It accepts, these ants in this colony are accepting this beetle as if it were a nestmate. And you find these beetles nowhere else except inside ant colonies. So they are evolutionarily specialized on the ant colony niche, okay? And the entire head, in fact, is kind of transformed into this straw, which is specialized for feeding on liquid food regurgitated to the ants. Now, How is the beetle able to do this? Well, it all depends on chemical trickery. So if you look at this part of the beetle's abdomen, you can see these orange tufts of hairs. You see the ants here licking this part of the beetle's body. These are called trichomes. They're these kind of biological candle wicks, which are conducting chemicals secreted by glands either side of the abdomen here. And these are exerting essentially a chemical mind control over the beetle. So the ants find whatever is secreted by these structures very attractive. They'll come up and feed on this part of the beetle's body, and it overrides the innate aggression of the ant towards other organisms and causes the beetle to be accepted inside the colony. Okay, so it's fooled them through chemical deception, and it's behaviorally assimilated into the social fabric of the ant colony. The ants love these beetles so much that the beetles have evolved all of these kind of adaptations to make it easier for the ants to accept them into the colony. So this part of the beetle's body is perfectly fashioned to fit the ant's mandibles, its jaws, right? And they'll pick these beetles up and carry them around the colony, sometimes depositing them in the brood galleries where the eggs are. And even though this beetle's head is essentially a straw, its jaws are still sharp enough to pierce open ant eggs and suck out the insides. Okay, so they chemically manipulate the ants and they freeload off the colony, parasitizing it through this social behavioral and chemical adaptations. This way of life, living inside ant colonies through these deceptive strategies, is called myrmecophily, or ant-loving. Organisms that can do this are called myrmecophiles. And if you look across the arthropod tree of life, you see myrmecophily evolving pretty sporadically in most groups. So there's one genus of cricket, which is myrmecophilous. Okay, you can see it being fed by the ant here. There's a family of butterflies that have caterpillars, like this one here, which are these socially integrated myrmecophiles. Okay, and myrmecophily is this kind of incredible example of communication between different species. Somehow these organisms are able to evolve these social behaviors that break through the species barrier, right? They kind of learn the language of these other species, these social insects, so that they can live permanently inside their nests. But it's really within the beetles where myrmecophily kind of takes flight, and you see more origins of myrmecophily than probably the rest of the arthropods. And when organisms like beetles move inside ant colonies, the selection pressures which are acting upon them are very different to those which affected their free-living ancestors. And this is manifest often not only in behavioral and chemical adaptations, but anatomical ones too, to produce really, really striking changes in body form. And I'll show you a couple of examples right now that kind of compare some myrmecophile beetles to their free-living close relatives, to just show you how dramatic and extreme evolution can be in these organisms. So this is a ground beetle, a free-living species, generalist predator that lives down in the dirt, kind of ecologically unremarkable. Okay, and that's what this thing looks like. This is a myrmecophile version of this beetle, which probably about 60 million years ago wandered into an ant colony and evolved into this incredible organism here. You can see these structures here, these big Mickey Mouse ears. These are actually the beetle's antennae. So the 9 segments of the antenna here have all fused together and expanded into this gigantic disc-like structure. And the disc is decorated with little glandular units that secrete what we refer to as appeasement compounds that ants find really attractive. So this beetle will wander up to an ant colony. Ants will approach it aggressively, but they'll bump into its antennae and find themselves attracted to the secretion coming out from these glands. And they'll actually pick this beetle up and carry it in the colony. And once it's inside, it again will feed on the ant's brood. So it's this complete Trojan horse strategy that this beetle has evolved through these chemical adaptations. Here's another example comparing a myrmecophile to its free-living relative. This is a pretty typical looking Rove beetle, like the ones I showed you earlier. Free-living, generalist predator, lives down in the dirt. This is a version of this beetle which has evolved to live in Afrotropical driver ant colonies, which are extremely hostile, aggressive ants. And you can see what this beetle has decided to do, at least in evolutionary terms, is transform its entire body into this protective shield. Okay, so you can't see any of the appendages here. They're all tucked underneath here. Okay, the wings and the elytra, the wing cases, they've all vanished. They've degenerated evolutionarily. It's kind of covered in these spikes, and the cuticle itself is extremely thick. And what this beetle is doing is it's withstanding the hostility from the ants. It's not trying to socially integrate like the myrmecophile in the previous slide I showed you. This beetle is a persecuted intruder. The ants recognize it as foreign and attack it, but the beetle doesn't care because it's invested so heavily in this physical protection. And so this beetle and the previous beetle really kind of are these two contrasting strategies that exist for making a living inside an ant colony. You either evolve chemical and behavioral trickery to gain acceptance of your host, okay, or you invest heavily in defensive strategies. Your host may recognize you as foreign, but you can keep them at bay through physical or chemical defense strategies. Now, back to rove beetles. One of the most kind of dramatic examples of myrmecophily that you see in rove beetles is seen in species which have evolved to parasitize colonies of army ants. If you don't know what army ants are, they are a real thing. They are a group of ants that lack permanent nest sites and emigrate and swarm across the forest floor, largely in tropical ecosystems, feeding on other arthropods. And they can form colonies that are huge, like millions of workers, and they're really kind of the dominant predators of tropical ecosystems. If you are walking in the tropical forest and come across one of these army ant columns, like this one here, and stare at it for long enough, perhaps one in every 500 or one in every 1,000 of these ants will actually be a rove beetle that looks like this, okay, this so-called myrmecoid body shape. There's several dozen different genera of these incredible ant mimicking species. We think that the change in the body shape of the beetle to this kind of ant-like form is not visual mimicry, because army ants are basically blind, okay? But we think army ants use a lot of tactile cues, signals to recognize members of their own colony, in addition to chemical ones. And it's this that selects for changes in the shape of the beetle. So in the tiny mind of an army ant, when they interact with this beetle, it will feel enough like an ant to be accepted inside the army ant colony. So you can see in these rove beetles, there's this really complex symbiosis that's evolved in army ant associated species in particular. So from this kind of ancestral rove beetle morphology and behavior, these ones that kind of look and behave like ants have evolved. And there's been a controversy in the field about how many times this incredible symbiosis has arisen, whether this had kind of arisen once, which many people believe because it's such a kind of incredible biological phenomenon, or whether in fact it evolved convergently multiple times. So while I was a postdoctoral researcher before I came to Caltech, we kind of solved this problem by collecting these beetles from army ant colonies in different tropical regions of the world. This is work I did with my collaborator, Munetoshi Maruyama, and sequenced the DNA from these species, as well as free-living rove beetles from across the rove beetle tree of life. And this is the evolutionary tree that we produced from performing this analysis. And what you can see here is this incredible repetitive pattern of evolution. Okay, so I'm just going to walk you through this tree. You can see all of these lineages here. These are different species, and they all descend from a common ancestor at some point back in time down here. And all of the black lineages, they are species that do not live with ants, right? And have typical kind of rove beetle morphology, like these representatives here and here and here and here. Okay, but you can see over and over, a dozen times at least, they've transformed into these incredible ant mimicking beetles. Okay, each one of these, each one of these ant mimics has evolved independently of all of the others. Okay, so this is an example, really incredible example of convergent evolution. Convergent evolution is what we term a trait which has evolved in multiple lineages of organism independently in response to similar selection pressures. So the most obvious example of convergent evolution is the emergence of wings in flying animals. Okay, wings have evolved in pterosaurs, birds, insects, and bats. Okay, so in response to kind of taking to the air, they've all evolved the same solution, which is to evolve wings. That's a single trait. What we have in these beetles is an entire lifestyle, the symbiotic lifestyle where the morphology and their behavior has all evolved in concert. Each one of these independent origins is associated with a single genus of army ant in a different tropical region of the world. Okay, so they're incredibly what we can say call host specific. So what this means is every time a beetle like this or this or this has evolved to ecologically associate with army ants, its morphology and its behavior have all walked down the same kind of evolutionary trajectory to produce something which is pretty unique in the animal kingdom, this convergent system of socially complex symbiosis. And just to kind of emphasize how really ridiculous convergent evolution is in this group of organisms, take these two beetles here. Okay, this is one of these myrmecoid symbionts that lives with this species of army ant in South America. This is one of these beetles that lives in colonies of this army ant in Southeast Asia. And look how similar these two look to each other, right? They look almost identical to each other, but they each evolved independently from ancestral beetles that probably looked very similar to these here, which are their closest relatives. So there's incredible, predictable convergent evolution leading to really striking similarities in morphology. But I want to emphasize that it's not just the morphology which is convergent and repeatedly evolving in this group of organisms, it's the behavior too. So if you look at one of these beetles inside a colony interacting with ants, you'll see that they are treated like royalty. The ants will groom these beetles. They'll keep them kind of parasite free. The beetles will groom the ants in return. And these beetles are found here and only here. Okay. If you take these beetles out of the colony, we have no idea why, but they'll die within the space of a few hours. They're so seamlessly integrated inside colonies of these incredibly hostile ants, and they're completely obligately dependent upon them. And that's crazy because these are very, very aggressive, nasty ants. If I try and, socially integrate and try and make friends with army ants, this is what happens. This is a picture of my gardening glove after I put it into an ant colony, an army ant colony in the Peruvian Amazon. They're amongst the most hostile ants on Earth, and yet these beetles are so perfectly adjusted to life inside their nests. So in these rove beetles, we have this really crazy phenomenon whereby, free living kind of generalist predatory things with kind of quite boring ecologies have evolved into these incredible social symbionts with really radical morphologies, behaviors, chemistries. And this has happened not just once, but many, many times. Okay. And so if you're interested in this question, as I am, about how new kinds of ecological relationships evolve, you can really do no better than study these beetles because they're so adept at doing it. They are really the virtuoso group of animals at evolving new ways at interacting, communicating with other species. And we've been talking about ants so far, but these beetles have infiltrated colonies of other social insects too, in equally dramatic ways. So termites are kind of second on the list for being parasitized by these beetles. This is what a termite looks like. Okay, quite different from an ant. But remarkably, these beetles have evolved to infiltrate termite colonies too and start to resemble the termites. So believe it or not, this beetle here is a rove beetle that lives inside a termite colony. It's a so-called termitophile as opposed to a myrmecophile. This is the beetle here. This is the host termite here. Again, they are living down in the dark in these galleries with no kind of visual information at all. We think that the beetle's morphology is again evolving to kind of mimic tactile communication that these termites probably use. The crazy thing about these termitophile beetles is that they hatch out of the pupil, the chrysalis, looking basically like this, but then they're fed mouth to mouth directly by the termites, and they start inflating into these incredible kind of grotesque balloon-like creatures, which anatomically mimic their hosts. Okay, so it's an amazing example of kind of morphological transformation. And this has evolved multiple times too. So these are just 4 independent examples of these termitophiles that are scattered across the Rove beetle tree. Okay, so again, it's convergent evolution of myrmecophily, termitophily, something clearly very special about these Rove beetles that keeps predisposing them to evolve this way of life. We asked kind of how far back in ant and termite evolution we could find evidence for these beetles. So the earliest ants and the earliest termites that we know of that were probably social and formed complex colonies are found in 99-million-year-old Burmese amber. And when I was at the American Museum of Natural History in New York, we were exploring this amber deposit and found the first evidence that these beetles are actually present, probably parasitizing colonies of these early ants and termites. Okay, so this beetle here, Mesosymbion, is a genus that we discovered and named. Mesosymbion means short for the Mesozoic symbiont, and it's the earliest example we know of like a behaviorally symbiotic organism that lived inside social insect colonies. In this case, we think it's one of these kind of defensive morphologies. So it's got a really kind of expanded thorax that covers the head. The head faces backwards in this protective kind of fashion. The antennae are really thick, which enables them to kind of withstand being kind of bitten by mandibles of ants and termites. So morphologically, it looks like a symbiotic species. And it also belongs to a group which today is full of these myrmegophiles and termitophiles. So all of these pieces of evidence together, the morphology and the evolutionary affinity of this beetle, tell us that this is exactly what it was. Right at the dawn of ant and termite social behavior, beetles like this had already turned up to the party and were exploiting ant colonies and termite colonies as this novel resource. So we kind of take it for granted that ants are this kind of ubiquitous group of organisms, right? This kind of dominant social insect group that like carpet our planet. But ant dominance, the kind of ubiquitousness of ants, is a relatively recent phenomenon. We know this because we can look back in the fossil record all the way back to the earliest ants and measure the frequency of ants in these different fossil deposits. And what you see is this really striking trend where for the first kind of 50 million years of ant evolution, ants appear to be pretty rare. Okay, so this is the percent of insects that are ants in these different fossil deposits from the timing of the first ants about 99-100 million years ago. And you can see ants are far less than 1% of the total number of insects in these early deposits, but about 50 to 60 million years ago, they start to increase slightly in abundance. By the time you get to kind of 20-25 million years ago, this is Dominican amber, Miocene Dominican amber, ants are now 40% of all of the insects in this fossil deposit. Ants today in terrestrial ecosystems are up to 25% of total animal biomass, right? So they've exploded in abundance across planet Earth in relatively, short space of time, 50 to 60 million years. And this must have radically reshaped the biosphere, probably drove lots of insect groups to extinction. If I go collecting beetles from tropical forest leaf litter, this is Malaysian leaf litter, this is my tray and sieve here, my gardening glove, which I use to scoop up little bits of leaf litter looking for these beetles, most of what I get out is ants, right? They kind of completely blanket the tropical forest leaf litter. They extend up into the canopy. They are running the show in tropical forest ecosystems, and they, invade temperate ecosystems with equal ferocity. What you can see when you calibrate the evolutionary tree of Rove beetles. This is the previous evolutionary tree I've shown you now, but dated with different fossils, so we can provide a kind of time frame over the diversification of these beetles. You can see that these beetles are much older than ants, right? They go back right to the base of the Cretaceous, maybe even into the Jurassic, okay? And all of them were basically free living, these black lineages, for most of ant evolution. But when ants start to take off in terrestrial ecosystems in this kind of window here, the past 50 to 60 million years, you can see all of these lineages, these kind of convergent origins here. These are lineages jumping off from the kind of free living ancestral state to take up residence inside ant colonies. So as ants have kind of expanded to dominate the planet, these beetles have really got in on the act and have over and over again infiltrated their colonies. So what is it about these beetles that predisposes them to do this? What is really special about Rove beetles that have, kind of poises them to evolve into these symbiotic species? Well, like I mentioned, there's 400,000 kinds of beetle that we know of, and these Rove beetles are only 15% of that. They clearly have some kind of secret. And I'll tell you what I think that is. So remember, right at the beginning of this talk, I mentioned that Rove beetles have kind of an atypical beetle morphology. They probably evolved from beetles that look much like this one here, that have these long wing cases, these long elytra that protect the abdomen. Rove beetles sacrificed that. They evolved these short elytra, and that enables them to move rapidly through substrates, like I'm showing you in this video here. Okay, they have this body flexibility. It kind of turned them into snakes effectively, but it left the abdomen physically unprotected. And this was an Achilles heel for Rove beetles. Okay, and what Rove beetles have done to counter the lack of physical protection is invest instead in chemical defense, right? So at the tip of the flexible abdomen here, they've evolved this chemical defense gland. If you break off this segment, you can see this kind of bag of cells here that's a chemical reservoir that contains this yellow stuff, which is benzoquinone, which is a topical irritant, a really nasty chemical that activates pain receptors. Now, if you put one of these Rove beetles with an ant, this is what you'll see. You'll see the ant try and attack the beetle, but it can flex its abdomen around, blast the ant in the face with these benzoquinones, and save its life. So what we think has happened in Rove beetles is that as ants have kind of exploded to dominate the biosphere, they've selected for organisms like Rove beetles that could coexist with them, because the Rove beetles can chemically defend themselves against ants. This is probably the reason why there's thousands of species of Rove beetle and how they're able to infiltrate tropical forests that are ant-dominated ecosystems. Okay. Now, if you can chemically defend yourself against ants and create kind of an enemy-free space around you, you can also wander into an ant colony and profit from the resources inside. If you're a predatory organism, then ant eggs and larvae are kind of attractive things for you. And we think what's happened in Rove beetles is that right across the evolutionary tree of these insects, they're able to opportunistically break and enter ant colonies, feeding on the resources there. And from this kind of facultative, opportunistic beginnings, many lineages have decided, wow, this is a really resource-rich environment. I'm going to make my living here. I'm going to specialize on ant colonies. I'm going to evolve into a myrmecophile. Okay, and this is why we get this repeated evolution of myrmecophaly across the tree of life of these beetles. Now, when they transition to this symbiotic existence from a kind of free living one, what happens is that their chemistry starts to change to produce chemicals that better enable them to manipulate ant behavior. Okay, so one of the key kind of adaptations of these myrmecophiles are changes in chemistry. I'm going to kind of compare the beetle in the previous slide to the beetle in this slide, which is now a true myrmecophile. This is a beetle called Lomacusa. You can see as ants will approach Lomacusa will loop its abdomen round, okay, just like it's about to deploy its chemical defense gland if it were a free living species. But now what this myrmecophile does is it secretes something, one of these appeasement cocktails that the ant finds really attractive. It will drink from the back end of this beetle, okay, and this attenuates the hostility of the ant towards the beetle. Okay, so the ant will accept the beetle now as a nestmate. It will even regurgitate liquid food to the beetle. Okay, and it will even up Lomacusa, carry it inside the colonies and deposit it in the brood galleries. And once it's in, Lomacusa is in there, it will actually lay its eggs. And when Lomacusa's larvae hatch out of those eggs, they secrete a chemical that instructs the worker ants to feed the beetle's larvae preferentially over their own larvae. So this is incredible chemical strategy for brood parasitism that's evolved in these beetles, and it's all been afforded by this modification of the body plan that's opened up the abdomen for innovations in chemistry through evolution of these glandular structures and manipulations of those chemicals that the beetles produce from an ancestral defensive compounds to these compounds that manipulate ant behavior and enable the beetle to maintain a permanent presence inside the nest. So Rove beetles kind of are this amazing intuitive paradigm for understanding how new forms of ecological relationship evolve. Okay, and it breaks down to changes in both behavior and changes in chemistry. There are those changes in chemistry from ancestral defensive chemicals to these appeasement secretions that pacify ants, effectively forcing the ant to interact with the beetle without any hostility. And then there are changes in the beetle's own behavior, right? From ancestral defensive behavior, where it wants to run away or chemically defend itself, to one of true social interaction, where they actually seek out ants and interact intimately with them. Okay. And it's this transition from free living to symbiotic and these changes in behavior and chemistry that accompany it that my lab is trying to explore at a deep mechanistic level. And so what I've been doing since arriving at Caltech is trying to develop a genetic model approach to understand the evolution of this way of life in rove beetles. And a lot of our effort has been poured into trying to develop kind of our own version of the fruit fly, genetically tractable model rove beetle. The species that we pioneered is this organism here. It's called Dolotia coriaria. It is a commercially available rove beetle that you can buy and dump into your greenhouse, and it feeds on things like fungus gnats and thrips and other crop pests. This is kind of generalist predator. And I was a postdoctoral researcher working in a Drosophila lab, always with the kind of idea of moving back into rove beetles, which were these organisms that I've known and loved and collected since I was a kid. And I chanced upon this organism one night because I realized I found, I think, a kind of one of these integrated pest management companies was selling it. And I couldn't believe my eyes, because in the kind of context of the evolutionary tree of rove beetles, Dolotia sits here. Okay, it's firmly embedded within this kind of radiation of these beetles that seemingly at the drop of a hat are kind of predisposed to evolve into these wonderful socially symbiotic myrmecophiles. So Dolotia has the exact ingredients I'm interested in understanding, these kind of evolutionary starting conditions for evolving from a free living organism like it is into a symbiotic species. Okay, it's the kind of ground plan for which living with ants has selected. So we've done a lot of things with Dolotia. We've sequenced its genome. Okay, this is a genome sizes of some different insects here, including the fruit fly Drosophila. You can see Dolotia's got a very nice small genome. That is a desirable trait. That makes it kind of tractable. The fewer nucleotides you have in your genome, the easier it is to work with. We've developed tools for gene manipulation using things like CRISPR. So this is an example of the Dolotia larva here. This is a typical larva. You can see it's got these 6 legs, 3 pairs of legs. Okay, but you can see in this CRISPR mutant here, where we've knocked out a gene called distillus, which controls leg development, you just have these stumps here. So you can easily make mutations in the genome of this thing to study genes that you're interested in. We've also developed methods to be able to insert pieces of DNA into the genome of this beetle using piggyback transposable elements that kind of fluoresce in the abdomen like this. That tells you that you've integrated some external piece of DNA. And this enables you to build tools to do things like misexpress genes in cell types that you're interested in and that kind of thing. And so that elevates Dolotia into kind of a genetically tractable model system to study things that we're interested in. And those things that we're interested in are its brain and its neural circuits that enable it to kind of detect ants. And in the case of Dolotia, release this chemical defense behavior that I'm going to show you in more detail in a second. And we're interested in the gland itself. Okay, this is the gland inside the abdomen here, these different cell types that manufacture the beetle's defensive secretion, the benzoquinones and the solvents that those benzoquinones are dissolved into. And we're interested in both of these things really as a foundation to understand how they've been evolutionarily modified in the hundreds of lineages which have evolved from a Dolotia-like ancestral starting condition into these wonderful myrmecophiles. Okay, so I'm going to give you a kind of overview of one of the kind of areas of research we're interested in, which is the brain and behavior of these beetles and how they've evolved to recognize and interact with ants. So you can tether Dolotia to a treadmill like this one here, okay? And you can bring an ant in on like a linear actuator, and you can see the beetle will chemically defend itself as if it was just running in an arena and it, you know, bumped into the ant, okay? Now, this is really useful because it kind of really tightly constrains the interaction between these two species. It reconstituted this ecological interaction between them in a way that makes it very easy to measure. And so we use these kinds of platforms to ask questions like, how does the beetle recognize the ant? This is a very basic question. More specifically, what we're interested in is what are the sensory stimuli that the beetle is using to detect the ant? And if we can understand that in Dolotia, where it chemically defends itself, then we can compare that to a myrmecophilous species and ask, how has that changed? Okay, the fact that we can tether this beetle to a treadmill like this means we can use kind of machine vision techniques to quantify things like the beetle's pose, you know, how much it's flexing the abdomen, what its antennae are doing during this interaction. We can get really fine-scale quantitative behavioral data. Using landmarks painted onto the ball, we can actually also calculate the path and velocity of the beetle as it walks through this infinite landscape. And those are really useful pieces of information. So I'm going to show you a simple experiment here where Dolotia is walking along a ball for 4 minutes. And during minute 3, we just touch Dolotia with an ant, okay? Ant on the linear actuator. Okay, now what we can do is measure the flexing of the abdomen. And when you do that, you find that during this minute of stimulus presentation, when the ant is actually touching the beetle, it's flexing its abdomen a lot. So this is the angle, the flexing of the abdomen. And it doesn't matter if the ant is live or dead, and it doesn't matter what species of ant it is, it will flex the abdomen repeatedly, okay, as it's chemically defending itself. So you touch the beetle with the ant, and the beetle recognizes it as an ant, and it flexes the abdomen and chemically defends itself. What are the stimuli that the beetle is detecting from the ant that tells it that this is a threatening organism that I need to chemically defend myself against? Well, it turns out that an important component of that sensory information are the ants' cuticular hydrocarbons, the very chemicals that the ants secrete onto their body that are telling the ants, oh, this is a nestmate or this is not a nestmate. Okay, cuticular hydrocarbons look like this. They're these long chain alkane and alkene type compounds, some with methyl branches. If you take an ant and kind of dip it in hexane, these chemicals shed off, and you can run them through a gas chromatograph and separate them based on their size. And you get these really kind of complex profile of dozens of these compounds here that range from carbon 25 to carbon 35 in length. And this is the complexity of this profile that ants are detecting to determine whether somebody's friend or foe, member of a colony or not. Now, this is the kind of same data for Dolotia, where we're measuring the flexing of the abdomen during this minute of stimulus presentation. So the ant is touching the beetle here, and it's flexing its abdomen many, many times using the gland. If we look at how the beetle walks during this stimulus presentation, you can see it's kind of rooted to the spot. So this is kind of flattening where the beetle has been walking into 2 dimensions. You can see the beetle's wandering around, and it's wandering around, and it's wandering around. And suddenly the ant touches it, and it stops, it's rooted to the spot, and it's repeatedly flexing its abdomen, okay, to defend itself against the ant. Here's another example of that trace. Now, what we can do is, it doesn't matter if the ant is dead or alive, you'll see this pattern, but we can take that ant and hexane strip those cuticular hydrocarbons off, and we lose this behavior in Dolotia. So if you take one of those chemically stripped ants, even though the ant is physically touching the beetle can no longer detect those cuticular hydrocarbons, and you lose the flexing behavior. And even though it's kind of face to face with this ant, it keeps walking and it doesn't try and chemically defend itself at all. And so using this kind of approach, this beetle on the ball, this kind of treadmill, this really constrained experimental platform, we're able to kind of dissect the sensory space of the beetle and the specific cues, the information that the beetle detects that builds up this kind of representation in the beetle's mind that this is an ant and I need to chemically defend myself against it. And what we think is that the beetle integrates a few different kinds of information. There's the ant cuticular hydrocarbons, okay? They have to be physically touched. So the beetle needs, its antennae need to touch physically the ant, okay? And we also think that CO2 from the ant may be involved to instruct the beetle that this is an ant. So all of these things together, the beetle combines in its little brain, and it causes it to release the gland, flex the abdomen, and secrete these benzoquinones. Okay, so that's how things are in a free living beetle like Dolotia. Okay, that's what they're using to detect the ant. Okay, and this releases this chemical defense behavior. What about myrmecophiles? How have they modified their behavior in response to these signals? So the other thing that we've done since coming to Caltech is developing a system where we can study a myrmecophile counterpart to Dolotia. Okay, so in the Angeles National Forest here, there is a very common ant species, the Velvety Tree Ant. Okay, this forms huge colonies of millions of workers. I'm going to play you a video of two of my students extracting one of these colonies. You can see, you know, you just break into it and it's kind of 100% ant. It's really, really impressive. Okay, but it's in colonies of this Velvety Tree Ant, despite the kind of hostile nature, that you find myrmecophile rove beetles. Okay, so if you in amongst the chaos, you can see little things like this guy. Hope you can see that walking around here. Okay, I'll show you a video of what this beetle does. It's called Skeptobius. You put Skeptobius in an arena with the ant, and the behavior towards the ant is very different to that which you see with the free living species. It actually climbs on top of the ant, and the ant appears to be completely cool with this. Okay, what is Skeptobius doing when it's on top of the ant? Well, I'll play you another video that kind of shows this quite clearly. It bites onto the ant's first antennal segment like this, and then I'll let you try and kind of see this for yourself. It scrapes its feet against the ant and then smears its feet over its own body. Okay, I'll let this play for another couple of seconds. Hopefully you can see that. So it's avidly kind of scraping against the ant and then smearing its feet over itself. I have another video that shows you this very clearly. This is infrared and kind of slowed down. You can see those legs smearing stuff over itself. What the beetle is doing is it's taking the ant's cuticular hydrocarbons, it's smearing its feet against them, and then it's smearing those cuticular hydrocarbons onto itself. Okay, it doesn't make its own cuticular hydrocarbons. It's shut that pathway down. It's stealing them horizontally from the ant so it can chemically resemble the ant and achieve perfect social integration inside the colony. Now, if you take the ant and the beetle after they've done this grooming behavior, pull them apart and do that same gas chromatographic analysis, you can see that the CHCs, these cuticular hydrocarbons of the beetle and the ant, match each other perfectly. All right. So it's achieved chemical mimicry through stealing the ant's pheromones. And this is how it's accepted inside the colony. What is Skeptobius using to kind of recognize the ant? Why is it so magnetically drawn to it? Well, you can kind of reconstitute these interactions between Skeptobius and an ant in these little arenas, like this one here. And like, because the behavior is so reproducible, you can kind of have tons of these arenas and you, again, use machine vision and learning approaches to quantify behavior in this system. Okay. And so, when you put Skeptobius with an ant, within a minute, it's climbed up on top of the ant's body, right? And you can quantify the distance between the two insects over a period of hours. And you can see that Skeptobius is on top of the ant doing this grooming, CHC stealing behavior for a lot of time, right? We think it actually spends about 60 to 80% of its life grooming these cuticular hydrocarbons to maintain its chemical mimicry. If you put another insect in, really any other insect in the arena with Skeptobius shows no interest in it at all. Okay, so this is a hemipteran bug that's labeled with these blue dots, and Skeptobius is kind of whirling around like a washing machine, completely uninterested in this other insect. Okay, if you kind of look at the distance between these two insects, there's no interest. The bug's also not interested in Skeptobius. There's no interaction at all. So what is it about the ant that makes it different to the bug? Well, we think it's the ant's cuticular hydrocarbons, the very things that in the free-living Dolotia were aversive, repulsive compounds that cause it to chemically defend itself. So you can show this quite easily by, first of all, killing the ant. You can kill an ant and Skeptobius will climb up and do this grooming behavior. Again, you can see these long bouts of grooming when the ant is on top, the beetle's on top of the ant. Then you can hexane wash the ant. I should also mention here that you can just take a piece of an ant, like the abdomen. So Skeptobius grooming the abdomen, kind of like Sisyphus pushing a ball around. They're just magnetically drawn to really any piece of ant material. Now, if you wash the cuticular hydrocarbons off the ant's body, you lose this interaction, okay? And then you can reapply specific cuticular hydrocarbons back to the ant corpse and reconstitute the interaction. So again, you get these long grooming bouts. So it's the ant body surface pheromones, these cuticular hydrocarbons, which the beetle recognizes and triggers this social behavioral program, this grooming behavior, where the beetle says, I want some of that. I need to scoop up these cuticular hydrocarbons. I'm going to climb on top of that ant, take them, and then start to resemble the ant so I can get acceptance into the colony. And so that's what's happening in this video. When you see Skeptobius wander up there, it sensed those cuticular hydrocarbons. So what's happened during evolution, we think, in the case of these myrmecophiles, is that ants are producing these CHCs. These rove beetles have evolved to detect those cuticular hydrocarbons, and they interpret them as kind of cues associated with a threatening organism. Okay, that leads to defensive behavior. And what's happened in these many lineages of myrmecophile, which have evolved from this kind of ancestral defensive condition, is that they've reinterpreted these cues as attractive cues. Okay, these are things that the beetle wants to interact with, and they elicit these social behaviors such as grooming. Okay, so this is kind of an example of a behavioral reversal to the same stimulus, these ant chemical cues. Skeptobius is also plugged into other ant chemicals. It appears to eavesdrop on other ant chemicals too. So if you take a ton of ants, bulk extract all of the chemicals from them, not only do you get these cuticular hydrocarbons, but you also get another category of chemical called iridoids. These are these smaller polar compounds. Okay, and we think that ants secrete these from their abdomens, and they are the trail pheromones that the ants lay down as they form trails across the landscape. Okay, and what we're able to do is take these iridoid chemicals and paint them into arenas. And what you find is that Skeptobius will religiously follow these iridoid trail pheromones. Okay, and you can again, beetle will just kind of walk infinitely round in this circle, following these iridoids. You can again, you kind of multiplex this in many arenas and extract this kind of, trail following behavioral data. You can show that it's specifically these iridoids which Skeptobius is detecting, and also that this is an adaptive feature of the myrmecophile not seen in the ancestral free-living kinds of species. So this is Skeptobius, and this is the distance traveled circling in these arenas. And it's only when this polar fraction, these iridoids are painted in these arenas, that you see it walking and walking and walking around in circles. It won't do it for the cuticular hydrocarbon fraction. Okay, and then Dolotia, the free living species, won't follow either fraction. So if you put Dolotia in an arena, this is Dolotia here, even though these iridoid trail pheromones are painted in a circle here, it's just wandering nervously around the wall. You can see it kind of at the perimeter of this arena, this blue dot. And that's in contrast to Skeptobius, which is religiously following the circle. So Skeptobius is this incredible thing, right? It's kind of evolved to eavesdrop on the ant's chemical information. It's recognizing the CHCs, much like ants do to recognize their nest mates. In the case of Skeptobius, it's recognizing its host that it wants to groom and parasitize. And it's plugged into these iridoid chemicals as trail following cues. And this probably enables it to disperse between colonies, enabling gene flow across the landscape. So what we have here is the kind of reinterpretation of chemicals in the environment by the myrmecophile beetle, okay? And it's probably the case that this has evolved convergently to many, many times in probably the majority of these myrmecophile lineages, this reinterpretation of ant cues as things that are attractive, things that these beetles want to follow, things these beetles want to plug into, and enabling them to maintain a permanent association with their hosts. And so now we understand this at a behavioral level. The future of my lab, or at least one of the kind of dimensions of that, is exploring the underlying neural circuitry that enables the beetles to detect and respond to these cues. And so one thing that we're trying to do now is integrate our genetic technologies into these beetles to be able to image from their brains while they're interacting in real time with ants. So you can see here, this is Dolotia walking on a treadmill. And what this kind of gray block thing here is a 2-photon microscope headpiece. Dolotia has had its brain opened up and dissected. So you can see this white material here is the beetle's brain. You can perform a surgery on this beetle and it will still retain its defensive behavior when it's interacting with an ant. And what we're trying to do in this beetle is introduce genetically encoded calcium sensors, which signal when certain neurons are activated. And by doing these kinds of experiments, we can start to pinpoint differences in neural circuitry between free living and symbiotic species, which are responsible for social behavioral evolution in this system. Okay, final slide. I just kind of want to sort of summarize what we're doing and why we are doing it. So, you know, this simple beetle ant system that we can kind of bring into the lab, what we're really trying to ask here is, how does one species of organism evolve to recognize and interact with another species of organism? And how did this ability evolve? Okay, now by studying a tractable system that we can explore in the laboratory and in the field, we are shedding light on this much more general phenomenon that kind of glues the natural world together, which is the emergence of interactions between species. And by dissecting this kind of this circuitry and these behavioral kind of evolutionary differences between species, we're finding these general principles that likely apply to other contexts where species have evolved new kinds of interactions. And I will stop there and thank everybody in my lab, in particular, Julian Wagner and Tom Narragan, who performed some of the work with Skeptobius, David Miller, who has kind of pioneered CRISPR and transgenesis in these beetles, along with Mina and Hannah, and also Sheila Kitchen, who has developed a lot of the genomic resources that are kind of the foundational for my laboratory's research. And I'd like to thank all of the bodies and foundations and Caltech internal centers that have funded my work since arriving at Caltech in 2017. And I will stop there, and I'm happy to take any questions from you. Thank you.
That's great. Thanks, Joe. This has been an awesome presentation. You guys can use the Q&A button at the bottom to ask your questions. I already see quite a few in there, so we'll just jump right in. Got a question here that says, do rove beetles also exhibit brood parasitism?
Yes, so I gave one example of that in the case of Lomacusa, where the ants are tricked into raising the offspring of the beetle. And that's just that. Lomacusa is one of the best studied myrmecophiles, with some history of European entomologists studying it. And I'm sure there are other instances out there, but for sure that's like a kind of poster child of brood parasitism. And like what's cool about it is it's got this kind of additional chemical manipulation thing worked in there, where the some secretion from the larvae causes the workers to preferentially feed the beetle's larvae over their own ant larvae. So that's amazing brood parasitism.
Great, great. Got another question here. It says, are there also cooperating species models, e.g. coral/fungus? What are the trade-offs in cooperating versus parasitical relationship? Under what conditions does one type of interaction work better?
That's a great question. So why stay a parasite when you can kind of be a mutualist and cooperate? So I would say that, in Rove Beetles, what we have here is this group that ancestrally were predatory. Okay, so the kind of motivation for getting inside the ant colony is to, exploit the colony, feed on, the brood and things like that. And really what myrmecophiles are glorified predators. Okay, and you have this kind of socially parasitic life history, which is just predation with some kind of chemical and behavioral bells and whistles attached to it. Why does that not move towards something mutualistic? Well, I would say most mutualistic relationships are really just sort of two-way exploitation rather than one-sided exploitation. And they probably have their origins, at least often, in ancestral parasitic relationships. And so you might find in these beetles, as in particular, some of the more ancient lineages, that they've evolved to be slightly less burdensome on their hosts and may actually be supplying something in return to them, even though they are still parasitic. So some of these lineages of beetles, for example, Skeptobius, through grooming the host, we actually think, in addition to stealing the cuticular hydrocarbons and achieving mimicry, it may remove surface parasites and pathogens from the ants. So it's not like a complete negative to have these beetles in the nest. There may be some actual positive attributes to having them around. And for all we know, nobody's ever quantified the impact of these beetles on a kind of colony by colony basis. It may be that it's possibly a zero-sum game for some of these lineages, the very ancient ones, which, if they haven't undermined their host, they found some way for long-term evolutionary persistence, maybe because they are contributing something back to the ants. Our interpretation, by and large, naively, is just that they are socially parasitic. And I think the majority of these lineages, that's probably true.
Yeah, that's super interesting, because there's been a couple other questions I see, or I've been noticing in the chat, that kind of relate to that. And one of them is coming up here. It's a two-part question. One says, besides secreting chemicals the ants like, is there anything the beetles do for the ants that is actually useful or essential for survival? Like what is it that they're doing for the ants is some of the questions I've been noticing.
Yeah. So yeah, I guess that fits with the previous question. Another thing that is potentially beneficial is some of the myrmecophiles are not kind of so trophically specialized on the ants, and will also feed on other things inside colonies, like mites, some of which may be detrimental to the colony. So again, it's more of a complex picture where a lot of what the beetle does is probably negative, but some of what it does may be kind of positive, including like spring cleaning and parasite removal. Some of the secretions that the beetles produce, we don't know what those are chemically, but the ants are absolutely obsessed with them. And for all we know, they actually may be, nutritious or something like this.
Yeah.
It's such unexplored biology that we simply don't know. And it'd be wrong to say that there's no, enhancement for, feeding on these secretions.
Yeah, absolutely. And kind of the second part of that question says, along those lines, if you take the beetles away, what would happen to the ants?
If you take the beetles away, so we do this in the lab, and we tend to find that like the ant colony does fare better, right? So it's kind of like a net negative probably having these beetles. But I have to emphasize that, we study kind of a model's example of this that we're able to kind of bring into the lab and study and like do field experiments with, because it's like a local example of this. But there are so many examples of this that there's probably not one rule. And there may be other instances where the beetles are kind of beneficial. There's one, actually, now I come to think of it, there's one unpublished observation here, which I think people asking these kinds of questions will find really interesting, which is that there was a species of myrmecophile that lives with a certain ant, species of ant, and somebody did an experiment where they put like 5 ants in all of these tubes, okay, and didn't feed the ants anything, but kept them damp enough. And then in half of those tubes, they put like one of these myrmecophile beetles. And in those tubes, those worker ants, the ones that had the single beetle, they all lived about twice as long, right? And so the hypothesis there is that maybe these kind of appeasement secretions like calm the ants down and like, you know, pacify them and just make them more chill. So, or through things like grooming behaviors, you know, they're, you know, maybe removing pathogens or something like this. But it's quite a striking result. It's never been, you know, repeated. It's something that we really want to do. But like that tells you that it's more complicated than just straight up parasitism, at least in some cases.
Yeah, no, that makes a lot of sense there. Another question coming in here, it says, where do the beetles living with ants go to reproduce?
So we know this in the case of Skeptobius, the grooming beetle, is kind of amazing. You know, they seem to cluster around ants that they're grooming, and they'll actually mate on top of the ants. So, you know, they've disguised themselves as ants. They're like, you know, they're laughing all the way to the bank, basically. So, you know, that they can mate with each other while one of them's grooming an ant. It's just like incredible. And they seem to kind of cluster around single ants and sort of use them as islands for lots of the kind of intra-specific stuff like mating, male-male aggression, those kinds of things. They happen in these kind of like clusters where there's one ant at the center. So all of the kind of Skeptobius' own reproductive biology is kind of still has to happen, and it seems to happen around kind of clustering on these islands that are, you know, these single ants.
Very interesting. Mating on top of the ants. We got another question here. It says, is it possible to speculate that ants and beetles were competing for food in the Cretaceans and therefore beetles evolved to steal from the ants?
Interesting. I would say unlikely, because ants probably were already this huge ecological force to be reckoned with. You know, they outnumber the beetles, you know, by orders of magnitude. I think what was probably the case was that, like, you know, ants just kind of started to, you know, proliferate in terrestrial ecosystems, and their colonies were, you know, just these concentrations of resources. You know, ants control the populations of all the other arthropods in the environment. They, you know, feed on nectar from aphids and extrafloral nectaries from plants. They harvest seeds, you know, and they transform that into new ants, you know, through brood and eggs. So there's a huge amount of kind of, you know, energy stored inside an ant colony. And if you can get your foot in the door and keep the ants at bay, like Rove beetles can do, then the payoff is huge. And so that I don't think competition, I think it's exploitation of this resource-rich niche, the ant colony, that explains this.
Fascinating. Got another question here. It says, is there any evidence of an ant evolving any defense to the beetles?
That's a great question. And no, there's not any evidence. But I would say, like, you know, in general, the stringency and like the kind of efficiency and reliability of ant nestmate recognition systems probably exists because, you know, to keep out intruders, right? And it's probably, you know, there's probably like this kind of co-evolutionary warfare between things like myrmecophiles selecting for ever better, you know, reliable nestmate recognition systems in ants. So, you know, presumably the pressure imposed by these beetles is one of the things which has driven the kind of chemical complexity of these CHC profiles that makes them very hard to mimic. through indulgent and, manufacturing profile like that yourself. And that in turn is selected for things like grooming behavior. So you just steal the entire profile. Yeah.
Yeah. That's great. These are some great questions. We got
awesome questions. Yes.
Yeah, no, our audience loves asking questions. This is the best part. We got another one here that's actually pretty interesting. It says, is it possible to weaponize the chemicals used by rove beetles as a way to fight ant or termite infestations, perhaps by attracting them to some alternate location? Or alternatively, would it be possible to weaponize the beetles themselves by allowing them to carry out their infiltration of the ant colony?
Yeah, this is an awesome question because, ants include some of the most invasive, nasty, organisms that, plague the modern biosphere. So for example, in Southern California, where I am, the Argentine ant has turned kind of LA County into an insect desert, right? It's just annihilated everything else. I knew it would be great if there was some kind of biological approach to targeting invasive ant species, which, wreak havoc on many ecosystems. And so one of the things that my lab is interested in doing is kind of taking the free living beetle, which is just a generalist, right, and trying to evolve it, engineer it into a specialist that could target something like Argentine ants. So we're interested in understanding the chemical ecology of ants in such a way that you might be able to engineer the attraction of this beetle to a specific ant species, in a way that would enable it to infiltrate the colony and undermine it. So yeah, you could potentially use rove beetles through this kind of approach, learning from the natural world and what rove beetles do so spectacularly to design strategies like this. That's a great question.
Yeah, that's super interesting. I have a question here. It said, would the myrmecophile beetles respond the same way in the treadmill experiment or otherwise if you covered some non-ant surface in CHCs?
Yeah, so can you put a dummy object, to sort of, I mean, those are the kinds of experiments that we were interested in doing, to try and really deconstruct the ant stimulus into it's just, the necessary and sufficient components. We think that the CHCs are critical, but they're not, but they're necessary, but they're not sufficient. There are these other cues, including physical touch. We don't actually know yet if kind of the shape of the ant or some other features of its cuticle are necessary for its perception of the ant as being kind of a threatening organism in the mind of Delotia. But yeah, that's the beauty of having that kind of system where you can reconstitute it. You can ask exactly these kinds of questions, but getting kind of granularity on the kind of shape of the stimulus is not something that we've done yet.
Yeah, these are awesome questions. We have a couple more here. One says, how do rove beetles change their appearance to better suit their environment? In the case of termitophiles, do they simply become more stout, or do they fundamentally change aspects of themselves, such as expressing termite proteins?
Oh, I see. So, I mean, that happens evolutionarily. It doesn't happen. So I guess in the case of the termitophiles, yeah, there's this kind of growth of the body that happens after they hatch out of the pupa. And I guess I need to clarify that. So those beetles, when they hatch out of the pupa, the insect has these hard body segments that are called sclerites, and they're connected by intersegmental membrane. And those termitophiles have a ton of intersegmental membrane. So that enables the body to inflate as they feed. And then they become these kind of expanded balloon-like, beetles. So, yeah, they can just accommodate a huge amount of kind of, food that gets turned into internal fat body and tissue like that expands the body. But they have enough flexibility because of this excess intersegmental membrane that enables them to inflate. Hopefully that clarifies why that question was asked.
Yeah, that makes sense. Here's an interesting one. It says, given that some beetles possess the ability to fly, are there any species that parasitize and or coexist with invertebrates?
I mean, ants are invertebrates. Do you mean vertebrates, maybe?
Probably. I'm just reading the question from.
Yeah. If it's vertebrates, yeah, there are some species that live on mammal fur. There's a whole group, this genus Amblyopeusa, which lives on, like, in rodent burrows and actually lives on mammal fur, and is actually beneficial for rodents because it will feed on mites and other parasites that are also living in the fur of mammals. So rove beetles are kind of ecologically amazing. They're just so diverse in their kind of life strategies. And like, myrmecophily, termitophily is like kind of an extreme manifestation of this. And they've evolved that many, many times. So there are all these other kind of quirky lifestyles that they've evolved, including, commensalism with some vertebrates.
Yeah, that's there's another question that kind of actually pertains to that. It says, do the beetles protect the ant colonies from other invaders?
So I don't know the answer to that. I would say that, sort of echoing what I mentioned earlier, some of the less specialized beetles will also feed on other things inside the nest, which might include other intruders like mites or springtails and things like this. So yeah, and to some extent, they're not like, they're just kind of grazing on other microarthropods inside the nest, which the ants also probably don't want to be there.
Interesting. Yeah. That's another question actually also kind of pertains to that. It says, are beetles the only species that live with ants and become predatory?
No. So, the myrmecophily has arisen, many, many times in rove beetles, a large number of times in other beetle groups, but less so than in rove beetles. But there are other origins of it in other groups of arthropods. So there are flies that are kind of socially parasitic myrmecophiles. There are those butterfly caterpillars I showed you, which are basically parasitic, which is incredible because most butterfly caterpillars just feed on plants, and they've actually evolved to be predators that live inside ant colonies. There are crickets. There are certain like hemipteran bugs that do this. There are millipedes. There's even a snail that's known to be myrmecophilous. So it's like this crazy phenomenon that's like, every time you see it, it's like the most amazing kind of natural history, and it's evolved in many groups.
Wow. Yeah, it's across all of them. That's crazy. I have a comment and a question here. It says, great presentation, thanks. You mentioned general principles you are finding. Can you give some examples, even if they are preliminary?
So yeah, I mean, in general, what we want to understand are things like, how does the nervous system of an animal recognize, another species of animal? And that's not something that we know very much about. And so by kind of understanding it in the really kind of simple case of Dolotia recognizing an ant, that probably gives you a logic which applies for every other species of organism that has to navigate the natural world filled with other species. And then, the kind of counterpart to that is understanding how that, nervous system has evolved to kind of, generate social behavioral interactions between species, and that's not something that we know very much about, like how social evolution happens through changes in neural circuitry. And so by comparing Dolotia to something like a myrmecophile that socially interacts very intimately with ants, gives you some insight and general kind of understanding of that. And in this case, we think that actually, the myrmecophile and the free living species are both detecting ant cuticular hydrocarbons. One of them, the free living species, thinks, oh, those are nasty chemicals, I need to chemically defend myself. And what the myrmecophile has done is say, hang on a minute, those are attractive things, I need to kind of groom this organism, or like, get it to feed me mouth to mouth and socially interact with it. And so that kind of what we refer to as a behavioral reversal or a valence reversal, that's really fundamental kind of neuroscience to do with innate behavioral evolution. And, there's kind of really profound example of it in these rove beetles. So answering those kinds of questions probably has real broad applicability to, all of the rest of the animal tree of life.
Yeah, that's great. We are coming up here on the 6:30 mark, so I'm going to try and be conscious of everyone's time, but we do have a few more questions and we'll wrap up. One here says, is it possible to determine the impact of parasitic rove beetles on the health of ant and termite colonies in the lab?
Yeah, we've not really systematically done that, but there are kind of ways that we are in ways you can do that, and we are definitely interested in trying to do that. You know, as well as just kind of measuring, like, you know, the fitness of the colony and whether it's, you know, how much brood it's producing. We really want to understand the mechanisms by which these beetles may impose a cost on the ant colony. Like, why would it be that suddenly, you know, the kind of reproductive output of the colony or the queen, or, you know, the amount of food that's harvested would be reduced by the presence of these beetles, you know? And there are probably layers to the answer for that question, but we have not systematically done it. But the fact that we can kind of reproduce these interactions in laboratory setting gives you kind of the experimental control now to be able to do exactly this.
That's great. And then I think we'll wrap up here. This is a good one I saved, related to the Argentine ants you had mentioned earlier. It says, do the Argentine ants have any mimicophily, or is that how you say it?
Mimicophile, yeah.
Mimicophile beetles associated with them?
That is a great question, because if you look in the literature, the answer to that is nobody knows. Okay.
Yeah.
So, you know, like Argentine ants, this kind of super colony, and there's never really been any mimicophiles recorded with them. But in 2020, another entomologist actually working, I think he's at UCLA, Graham Montgomery, he discovered an Argentine ant colony near the port of LA that had a myrmecophile rove beetle living inside it. And that myrmecophile rove beetle is of a kind that's only ever been discovered and found in South America. And so that probably tells you that during the introduction of these ants, one of these myrmecophiles has actually kind of been co-introduced with it. And we're very interested in finding this myrmecophile again and studying its biology, because if there's one thing that can kind of make a living inside Argentine ant colonies, and, you know, potentially to the detriment of the colony, that's another possible tool in the arsenal for dealing with Argentine ants. But that's unpublished, kind of just very recent discovery.
No, that's super interesting that they could potentially be co-introduced. That would be
it was an incredible tropical thing. Like, I could not believe it was found near the port of LA. Like, and I think in South America it's only known from like a small handful of specimens, but, you know, it's walking around near in LA County. It's crazy.
Made it all the way up the coast. Wanted that LA weather, right?
maybe so, yeah.
All right, cool. Well, I think we'll wrap up here. Thank you everyone again for tuning in this evening, and thank you again, Joe, for taking the time to speak with us tonight. I didn't realize how interesting beetles were until tonight, so this was super interesting. I think all the attendees definitely found it fascinating based on all these questions we had. For our audience members, our next Café Sci will be coming up in June, so be on the lookout for any communications related to that event. And with that, have a great rest of your Thursday evening, and I look forward to seeing you all next month.